Colleghe e colleghi, cari amici dell'associazione Sindrome E.E.C, è un grande piacere partecipare all'apertura di questo convegno sulla malattia E.E.C, mutazione di P63. È un momento difficile per il Paese, è un momento nel quale l'importanza e l'impegno congiunto di tutti, medici e cittadini, nel difendere il proprio diritto alla salute è sotto gli occhi di tutti, è sotto gli occhi di tutti nel momento in cui una grave crisi sanitaria sta interessando l'intero pianeta. Ma quello che ci insegna la grave crisi sanitaria è quello che le associazioni come la vostra e la nostra università portano avanti da tempo, e che ogni aspetto della salute dei cittadini va tutelato, e va tutelato affidandosi alla scienza, una scienza rigorosa che affronti il cuore dei problemi e con grande fatica, grande rigore, ma l'unica strada che ci permette realmente di arrivare a un risultato, provi a trovare sempre nuove strade terapeutiche di grande successo. E allora noi siamo orgogliosi che la nostra università, anche nella sindrome E.E.C, grazie al Dipartimento di Medicina Molecolare che svolge ricerca su questo argomento, sia in prima linea, perché siamo convinti che attraverso questo impegno noi rispondiamo ai valori dell'università. I valori dell'università sono quelli di sostenere la scienza, una scienza libera e coraggiosa come quella del nostro grande studioso Galileo Galilei, ma rispondiamo anche a quel bisogno di giustizia che ha sempre animato la nostra università, e quindi dare diritti a tutti, affrontare e risolvere le difficoltà di tutti, in nome di quella patavina libertas che è nel nostro motto e nel nostro cuore. Allora tutte le malattie sono importanti, quelle che colpiscono come la pandemia il mondo intero e quelle che hanno pochi pazienti, ma pazienti che hanno il diritto di essere tutelati nella loro salute, di avere qualcuno che studia per trovare nuove cure per la loro malattia. E allora attraverso l'impegno per la sindrome E.E.C, che poi è il grande impegno che il nostro Ateneo ha per un gran numero di malattie rare, noi vogliamo essere una spinta, e una spinta non solo verso questi pazienti e verso queste famiglie, ma anche verso l'idea di una scienza libera che si impegna duramente e con passione a favore della salute di tutti i cittadini e tutte le cittadine.
Buongiorno a tutti e bentrovati cari amici e cari partecipanti. Sono Cristina Bolzonella, apro in italiano questo importante appuntamento dedicato a ricerca e confronti, giunto al quarto appuntamento. Innanzitutto un saluto in italiano perché siamo a Padova, la nostra associazione è nata a Padova ed è rappresentata da me e da Giulia Volpato qui al mio fianco e da tutto lo staff chiaramente che ogni giorno lavora in italiano da Padova nel mondo. Quindi una piccola premessa prima di passare alla lingua inglese, che sarà la lingua ufficiale di questo incontro, per dare l'opportunità a tutti di seguire non soltanto il nostro lavoro, ma il lavoro che è stato svolto in rete da tantissimi professionisti che finalmente siamo riusciti a mettere insieme in un momento epocale. Quindi il webinar va ovviamente a toccare un punto importante ed è stato onorato anche dall'apertura del rettore dell'Università di Padova, il magnifico rettore Rosario Rizzuto, che sta seguendo il nostro operato e ha messo a disposizione anche i vari dipartimenti dell'Università di Padova proprio per incentivare maggiormente non soltanto il contesto della ricerca, ma anche di tutto l'impatto della situazione del rapporto psicologico e dell'implementazione e della qualità della psicologia applicata alla qualità di vita e allo sport. Saluto anche e ringrazio tutta la direzione che ha coinvolto Padova Capitale del Volontariato 2020, che affianca tutto il nostro operato e che ha permesso anche di valorizzare e registrare questa importante azione. In che senso registrare? Registrata perché rimarrà nel tempo. Padova Capitale del Volontariato 2020, ovviamente in un momento difficilissimo, una pandemia mondiale che ha però valorizzato l'indotto del web ancora di più e ha quindi espresso al massimo tutte le potenzialità che i collegamenti da tutto il mondo, sia dei partecipanti ma anche dei relatori eccellenti, sono stati messi al centro. Quindi noi stiamo parlando da Padova, oggi 26 novembre 2020, abbiamo una quantità di professionisti eccellenti che garantiscono con il loro lavoro quotidiano la qualità e le risposte che sono state, che vanno date ogni giorno e soprattutto quello che voi avete inviato come richieste perché il sapere e avere delle risposte certe è fondamentale, soprattutto quando si parla di qualità di vita. Altre organizzazioni quale l'ULS e la Regione del Veneto che ovviamente non soltanto affiancano ma anche fanno il monitoraggio sempre delle nostre azioni. Essendo in un ambito sociosanitario questo è estremamente importante. La nostra organizzazione lavora dal 2009 nel territorio e nel mondo, dal 2012 ha comunque espresso la volontà di una rete soprattutto dedicata ai giovani, parla un linguaggio giovane, ma ancora di più punta a creare una grande comunità. Grazie a Ricerca e confronti, quarta edizione, possiamo veramente parlare di innovazione di rete, innovazione di contatti, innovazione di qualità. Pensiamo anche che nell'ambito della ricerca e del contesto clinico e delle linee guida si sta cercando di creare in Europa un specifico punto dedicato alla Sindrome E.E.C e al GNP63 e sto parlando degli Ern-Skin, al quale anche la nostra associazione afferisce e in modo particolare rappresentata dalla vicepresidente Giulia Volpato, dottoressa Giulia Volpato, la quale ne è anche referente per i pazienti in Europa. Quindi, cari amici, a questo punto devo dire cari amici perché non posso parlare di pazienti, posso parlare di una rete di amici e una rete di contatti veramente importante che vede da una parte i pazienti e le famiglie e dall'altra degli eccellenti operatori specializzati che siano chirurghi, ricercatori, medici, operatori. Questa quindi grande comunità, benvenuti al nostro incontro di oggi 26 novembre 2020, che vedrà da questa mattinata sino alla sera una serie di importanti tavoli di lavoro e durante i quali saranno lette le vostre domande, ma anche saranno acquisite ulteriori domande sia dall'Italia che dall'Europa, alle quali poi risponderemo. Ho quindi il piacere di passare la parola per l'apertura ufficiale dei lavori e in lingua inglese alla vicepresidente, dottoressa Giulia Volpato.
Thank you, Cristina, for your Italian presentation, and welcome to all of you at the third international meeting on EEC syndrome and P63 gene, organized by the P63 EEC Syndrome International Association, with the collaboration of lots of institutions, national and international institutions. This year is a really important years because of the COVID epidemic moment that all of us are living. So thank you for your participation to this meeting in this very difficult moment. Today and tomorrow, we will speak about the P63 gene in all these aspects, from the research to the genetics, from the plastic surgery to the psychological effects for patients and families affected by the P63 mutation. This year, we collaborate also with European Federation of Rare Diseases, so Eurordis. Thank you to Eurordis for your participation. And of course, we are participating also to the Ern-Skin international networks. So we will have a session dedicated to the Ern-Skin and focused by the guidelines of P63 in which we are active parts. So today we start our meetings with the research, and I would like to introduce the first speakers of the meetings, that is Dr. Enzo Di Iorio. Enzo, thank you.
Good morning. Thank you very much for this introduction. I thank not only Cristina Bolzonella and all association, P63 International Association, and not as last, the rector of the university. And I thank all my colleagues that are guaranteeing the continuity of this important study on EEC syndrome, a complex pathology. The term means ectrodactyly ectodermal dysplasia clefting syndrome, and we are studying in particular for the eye condition, the severe corneal problematic that can induce the blindness for these young patients. About our background, just a few words to introduce myself. I'm a doctor in biology and specialized in genetics, and I'm studying this pathology probably now 15 years of age. And about our background, we are expert in epithelium stem cell, in particular for the cornea. And cornea is an intriguing model to study stem cells because there is a physical segregation between the progenitor cells, so stem cells, and adult cells. And in these young patients affected by EEC syndrome, there is a severe condition that premature decline the stem cells of the cornea. It means developing in the ears a dysfunction of the cornea, and the consequence of this pathology are inflammatory disease, but not only, but there is a conjunctivalization. So the migration of a conjunctiva onto the central part of the cornea. This consequence induced the blindness, a progressive clouding of the opacity of this and the loss of the vision. The limbus, so, is defined the home of stem cells, characterized by this stromal invagination, and in a normal eye, all stem cells are localized exactly in this area, in this portion defined as palisect of Vogt. What happened in E.E.C syndrome? There is a progressive decline, disappearance of this structure, and at the end means having the loss of stem cells. But stem cells are essential to maintain the normal tissue homeostasis of the cornea, are essential to guarantee a long-term self-renewal of the continuous regeneration of the cornea epithelia. But when this occurs, when these stem cells are all destroyed, so the migration of the conjunctiva occur. So we are studying stem cells over this patient. Stem cells are essential to generate, to give rise to whole progeny of differentiated cells and generate adult cells. And in our background, we study in particular way the limbal epithelial stem cell, exactly the cells that are defective in E.E.C syndrome. We demonstrate that only stem cells are capable, when cultivated in vitro, are capable to generate this big important clone defined as holoclone that represent the maximum expression of stemness using in vitro model. But these are important aspects in regenerative medicine when the aim is to produce in vitro a graft, a sheet containing stem cell of the patients that we want to use to treat and correct a particular condition, such as happened in E.E.C syndrome. And well, E.E.C syndrome is a severe condition that involves limbus, that involve the epithelium stem cell, are known different pathology that are restricted on the cornea or on the limbus. But anyway, the E.E.C syndrome caused by mutation in P63 gene compromise the stem cell function of the cornea. And about this is the conjunctivalization. So this is the last event that usually observed in these patients. So the children the children probably are normal limbus. In fact, are predominant during the early ages of the history of these patients. Other factors are ectrodactyly, syndactyly, and other oral facial malformation, but in adult, the predominant aspect is the eye condition. They lose the vision ability, and we want to we want to help this patient and to rescue this severe condition that is now irreversible, and there is not exist a treatment, feasible treatment, and at the same time useful to correct. But what happened if we want to perform a corneal transplantation, a keratoplastic? So this is the normal treatment that we used to apply for all conditions when the cornea is affected by, as well as keratoconus, when there is a shape alteration of the structure of the shape of the cornea. But not is sufficient, is not enough in E.E.C syndrome, because when we have the stem cell defective, so we have to correct the gene P63, the genetic of the P63. And the solution could be cell therapy. In fact, cell therapy consisting into amplification of autologous limbus stem cell in order to obtain a final product that, after is applied, is successful to correct the corneal condition. But now we want to, before of this application of this procedure, we want to correct the P63 gene that we well know, and the strategy that we are using are different, but in particular, after this characterization, where we demonstrate that in this patient there is an hypoplasia of the epithelia, so there is a thinner epithelia, so the absence of stem cells induce a severe defect in the stratification of these cells. And the strategy that we are using is based on sRNA. Allele specific sRNA is a gene therapy strategy capable to inhibit the expression of a mutant gene, and this is important to obtain a normal, correct phenotype of these cells. And we are developing an important high drop containing these molecules, very small molecules that can enter in the cells, in the stem cells, and can correct the stratification and guarantee the normal self-renewal of the cells. And in vitro model that we published 2 years ago, we demonstrate the ability of this molecule to induce the correction of the stem cells of these patients. Our findings are really promising, and we are really happy to briefly produce these drops that we want to treat patients, and we hope long-term treatment for the entire life, capable in order to maintain the stem cell function forever in these children. So this could be an excellent treatment to prevent and to reduce the probability and the risk to develop on this limbal stem cell deficiency and so the pathology typically observed in E.E.C syndrome. So we are performing this study bypassing animal model, but using particular innovative machine an instrument that can mimic the function of the entire eyeball and the cornea in vitro. And so using these methods, we can test the efficacy and ability of this molecule before the clinical trial, but we are ready to start. We are so available 6 patients, and we want to start this treatment in order to establish the successful outcome or not. I thank all, and in particular Cristina Bolzonella, a good friend in all this study, and the importance of the network all inside. E.E.C is a rare condition. 1 on 1 million is the incident reported in literature. So it means the important having this association that maintain this contact between scientists, and not only scientists, but business is also important, because the requirement money essential to perform this research. I mean the grant and the presence of the E.E.C patients that believe in this study. Thank you very much for attention.
So thank you for your presentation, and I give the word to the next one speaker, that is Dr. Caterina Mistero from the Change Naples University. Caterina, thank you.
Yes, good morning. I'm going to share my screen to show you my presentation, introduce research. Wait a second, because of course. Okay, so so thank you for the invitation. I work at Change Biotecnologie Avanzate, an institute in Naples dedicated to genetic research. And also, I'm a professor at the University of Naples, Federico II. And our work has focused on the gene P63 for a long, long time now. So, as you all know very well, P63 mutation causes a range of developmental anomalies, including ectodermal dysplasia, split end and foot malformation, as well as orofacial clefting and some other problems, including corneal problems, which are due to mainly lacrimal and meibomian glands alteration, but also defects in corneal stem cells. So so there are PCC3 mutation cause different disorders, which have overlapping features. So as I said, ectodermal dysplasia, cleft lip and palate, and defects of the hands and feet, which are not found in all the different syndromes, but they are specific for some, but not for others. And as we will see, this is very much dependent on the kind of mutation that is found in P63. So research on P63 has started in 1998 when the gene was actually discovered. Since then, it has 5,400 publications have been published on P63. So P63 is a very active research field with approximately 220 publications on E.E.C syndrome and 220 on E.E.C syndrome, which are the main syndromes that are under research. So just to tell you about the chronology of the discoveries that were done in for P63, as I said, in 1998, P63 was discovered, and actually was a very important discovery because P63 is actually a member of the P53 family, and P53 is a very important tumor suppressor gene, and it was thought to be alone. Instead, in 1998, it became apparent that it had a brother. So P63 was thought to be the little brother of P53, but as we will see later on, this is not actually the case. In 1998, a mouse model for the deletion of P63 helped us to understand what was the relevance of P63 in skin and in other stratified epithelia. Then still in 1999, it was discovered that the E.E.C syndrome is due to P63 mutations. In 2001, it was discovered that A.C syndrome is due to P63 mutations. And then there were a number of discoveries, including the fact that P63 is highly overexpressed, is very much abundant in squamous cell carcinoma, and I will get back to this. And then, as you can see here, it has been discovered that P63 is important for the skin, for differentiation of cells in the skin, but as well as for proliferation of these cells, for growth of these cells. And later on, several mouse models have helped very much to understand both the diseases as well as the physiology of P63. And so this is what we are going to see in this presentation. And then finally, in the last few years, we have been able to make a mouse model, an important mouse model for A.C syndrome, understand the mechanism through which P63 mutation alter development in A.C syndrome, and also to understand the general role of P63. But let's, take one step at a time. So first of all, as I said, in 1999, two important discoveries were made that suggested that P63 was actually a very important gene for epidermis, for the skin, and for many other epithelia, which are stratified epithelia. Stratified epithelia are those of the esophagus, of the oral cavity, of many other districts, actually, and are also, so P63 is important for all these tissues, but also for a specific stratified epithelium that is important for development of hands and feet, and also cornea and many glands. So as depicted here, so this is a normal newborn mouse, and this was the mouse in which the gene was completely deleted. And you can see that they look pretty different. First of all, there is a problem which I think it's better depicted here. So this is a knockout mouse during embryogenesis. This is a wild-type mouse during embryogenesis. You see the development of the legs and arms, and as well of the face. And you see here that no arms and no legs are made without a P63, and also that you see here there is orofacial cleft, so which originates at the end cleft and lip palate. So clearly P63 is a gene that is required for the formation of, for the closure of the palate, for the formation of the entire skin, and formation of the limbs, which are the feet and hands. Well, the upper limb doesn't matter. So, and you see here, this is an histology of the skin. This is a normal skin, and this is the skin in P63 knockout mice. Basically, there is no skin. So at the time when this discovery was made, I was just starting my own lab, and I had an experience in which I made in the United States, working on skin. So I thought this gene would be extremely interesting to study and to understand why this gene is so important, for the skin, such as the skin is not formed without it. In addition, at the same time, it was discovered that E.E.C syndrome was due to mutation in P63. So, of course, in humans, the mutation in P63 are much less severe than the complete deletion of P63 in mice, because here we still have some, so the P63 is not functioning very well, but it's still functioning. Here we have no P63, so this is the difference. All right. So then, you know, in time, it was discovered by us, by also many other labs that have worked on P63, that this gene is very important for many processes, which are cell proliferation, so growth of cells of the epidermis, of the skin, also stemness. So if you don't have enough P63, cells will not divide enough to make a normal skin. Cell adhesion, this is very important. So the cells have to, especially on skin, they have to adhere to each other, to stick to each other in order to be resistant to mechanical stress. With the P63 mutation, this mechanical stress leads to disruption of the cells, of cells attached one to the other, and therefore there can be skin erosion, which, as we are going to see, are very dramatic in E.E.C syndrome. And then finally, P63 is also very important for the development of stratified epithelia and for tissue identity, which means that P63 is a master regulator, regulates the entire program of epidermis, of the stratification in epidermis, by activating epidermal genes and repressing non-epidermal genes. This is because P63 is a transcription factor. Transcription factor means that it's one of those genes that regulate the entire program of epithelial cells. Another very, very important characteristic is that the P63 is essential to convert stem cells into epidermal cells. This is true in vitro, but also in vivo. So when the embryo grows, the presence of P63 in some cells, but not in other, will lead to formation of an epithelium. So this is also a crucial, so this is really a crucial regulator, basically. Another important point that has been a reason for a lot of research, and we're actually also working on this, is that the P63 is known to be overexpressed in squamous cell carcinoma. So in those carcinomas of the skin, the oral mucosa, the esophagus, and the lung, where actually in the lung, P63 is very poorly expressed, but when there is a squamous cell carcinoma, which is the typical lung tumor of smokers, P63 is overexpressed. So meaning there is a lot of it. And the fact that there is a lot of P63 forces the cells to grow much more than usual, and this is what is causing. It's not the only reason why we get the tumor, but it's one of the reasons that allows the tumor to grow. So clearly, with this in mind, we have to be very, very careful when we think about gene correction, gene therapy for patients that carry P63 mutation, because we do not want to alter the balance between in the expression of P63. We want to make sure that we correct exactly the amount of P63 that we want to have, but not overexpress it. Otherwise, we risk to end up with the carcinomas. So P63 is so the mutation in the P63 gene are causative of a malfunction of the protein, because as you know, our DNA contains a lot of genes. That these genes are then transcribed into RNA. This is the general dogma of life, which are then translated into proteins. And proteins are actually the building blocks that of the cell that are the functional blocks, so those that really do the job. So in P63, when the DNA is normal, the normal gene makes a normal protein. When there is a mutation, the mutation disrupt is basically causing either no protein, but this is not the case in the case of P63, or an abnormal protein. So the protein is functioning, but not functioning as well as it could. And therefore, you get a phenotype, an alteration. So the important point is that what is the function of this protein that is made? So we said it's making a protein, P63 is making a protein, P63 protein, which is a transcription factor. What does it mean? That binds to the DNA itself. So the protein binds to the DNA and regulates many genes, all the genes of the epidermis. So it's basically controlling when the DNA is transcribed into RNAs, and for all the genes that are expressed in the epidermis. So it has a very important function. So if you want to depict the P63 is actually doesn't work by itself, but it works in tetramers, four copies of the protein that stick together and bind to DNA in order to regulate expression of epidermal genes. I don't know if this is too complicated, but if you have questions, please let me know. So the protein structure is quite complicated, but we can visualize it as a string. And this string has different domains. Different domain of the protein that do different functions. It's just like a modules, there is a sum of modules of different function. And so one of the function here is binding to DNA. So transcription factor has to bind to DNA in order to make it allow transcription. So this region here and this is important for what I'm going to tell you in a second, is the binding of DNA. And this other region here, instead, as a sort of an unknown function. This region here allows tetramerization, so the 4 factors that come together. So you can see here that the different mutants actually are found in different portions of the protein. So for the E.E.C syndrome, most of the mutations fall in the DNA binding domain. Therefore, binding to DNA is not very strong. And this is why during development, when you need a strong binding, you get some malformation. In contrast, E.E.C, which is this other syndrome that causes instead a bleeding after birth and erosion of the skin, this is a very severe phenotype. In this case, the mutation cluster all in these domains that have unknown function. They are not completely unknown, but just to simplify, they have an unknown function. So in our research, we focus on the A-well syndrome or A.C syndrome just because this major phenotype, which is these devastating skin erosions, occur after birth. So it is possible to think of a therapy for either a pharmacological therapy or a gene therapy to fix the skin of these babies. Okay, let me skip this. So in order to study what is this syndrome due to, and what is the what kind of therapy we can design, we made mouse models recapitulating the disorder by making a single mutation in the mouse P63. And so what we found is that mice that have a mutant P63 have a cleft palate, exactly like in humans. They have abnormalities of the teeth, and also have problems with their skin that are very similar actually to the skin erosion that we find in mice, in humans, I'm sorry. And this is due to, this is a wild-type epidermis, an histological wild-type epidermis. You see here that basically, as I was telling you, because of the very important function of P63 to make cells stick one to the other, this epidermis is fragile, is not as strongly attached as in the normal skin. This occurs spontaneously, and this is why you get erosions. We also discovered that the mutation in the A.C syndrome cause the protein to be unstructured. And this leads to the formation of protein aggregates. So instead of binding normally to the DNA, protein aggregates float around in the nucleus and therefore are not functional. So let me finish this presentation by acknowledging my collaborators, many young, very enthusiastic people, some in the long-standing collaborator, Dario Antonini, who actually has been working on P63 with me since 2002. This work could not be possible if we were not in a very nice and organized scientific institute, Change, and more importantly, it would not be possible without the help of Teleton. Teleton Foundation has been instrumental to carry on the research in Italy, focused on rare diseases. And especially on P63, they have given a great contribution. And you can see here where, first of all, they have funded the research at the University of Torino, Milan, Trieste, Padova, Ferrara, Bologna, Modena and Reggio Emilia, basically everywhere, including our work. I have to say that without Teleton, I would not have been able to do all this. And you see they have invested 3.8 million euro on the P63 and study 3 different pathologies: EEC with us, EEC, and orofacial cleft. They've been funding 14 researchers, 12 research centers, and the results are up to 156 publications from 1997 to today. So this is really a great achievement. And again, without the Teleton, we couldn't have done this. So thank you very much for your attention. And I stop sharing.
Thank you, Caterina, for your presentation. We are receiving so much of questions, so we have a few to make to you. Please, Roberta, can you read the questions?
Yes, the first question is, what are the variants of E.E.C syndrome most studied by scientific research?
Okay, so this is actually a very interesting question because this is a work very much ongoing. It has become clear in the last few years that different mutations have different effects. And so we are now studying at the genomic level a number of mutations and discovering that they have a very different mode of action. So this will be a very promising studies in the future. Also, we have done a screening, a general screening to identify different EEC, even those that have not been discovered as yet in patients. And this is like an unbiased way of discovering mutation that may occur in patients but have not been seen yet. So definitely there is, again, a genotype to phenotype correlation in that each single mutation affects the binding of P63 to DNA in different ways. And we are studying the molecular mechanisms.
Thank you. The second question was, what do you expect from the new frontiers of P63 gene research? Just for E.E.C syndrome or something else?
As I said during the presentation, P63 is very important also for tumor research, for squamous cell carcinoma research. So many people are including us, are investigating toward this direction. In the last few years, it has been possible to study. So the basic research is completely revolutionized because we are doing everything by looking at the global picture rather than at each single gene. So the discoveries are ongoing and the field is very, very promising.
Thank you. The last. Yes. Do researchers and geneticists work together?
Yes, they do, of course. We actually researchers work together with clinicians and also with the geneticists to identify, really to be able to look into a problem from a different perspective. So one of the most interesting area of research that connects a researcher to geneticists is to understand why people with the same mutation have different phenotypes, different defects, I mean. This is a very interesting area of research because that means that because each one of us is different from other people, there are genes that modify there are modifier genes that impact on P63 as well. So we still do not know which genes impact on the P63 function and therefore on patient health.
Thank you again, Caterina, for your answer. I would like to remember to patients and families that if you have other questions, you can write us and you will receive an answer in our Facebook page during the next weeks. So I give the word to the next one speakers, that is Dottoressa Sabina Di Fresa, by the Genetic Department of the Treviso District. Sabina?
Good morning, everybody. I thank you very much, Giulia and TP63 Association, International Association, for this invitation. Let's talk about P63 gene and phenotype. TP63, acronyms for tumor protein 63, is a gene located on the long arm of the chromosome number 3 at the 3q28 locus of the DNA, where is the red line in the upper part of the image. In the middle part of the slide, we find a schematic representation of intron-exon structure of the gene, of the human P63 gene. Each gene in our DNA is responsible for production of proteins with different functions. And each gene is made up of a functional region, the coding region, exons, and unfunctional regions, the encoding regions, introns. In the image, you see introns in white, while exons, the coding regions, are colored according to the functional domain of the protein. As you see below in the slide, there are more than one isoform of TP63 protein that are protein variants, and are at least 6 for this type of gene. TP63 gene encodes a transcription factor, which plays a fundamental role in the development of the skin and of other tissues of ectodermal origin, and also on the development of orofacial structures and limbs. As you can see, P63 knockout mice exhibit development defect on eyes, on skin, and on limbs and yolk. Mutations in different domains of the gene may lead to dysplasia of the Hus and other clinical features with a significant match, as you can see in the right part of the slide. Regarding a clinical characteristic, the TP63 related disorders comprise these 6 overlapping phenotypes that are EEC3, ectodactyly, ectodermal dysplasia, cleft lip, cleft palate, AEC syndrome, the adult syndrome, sorry. The first individual affected by the first 4 conditions have varying combinations of ectodermal dysplasia, but also lacrimal duct obstruction, hypopigmentation of the skin, hypoplastic breast, and or hypoplastic nipples. And the possible signs of ectodermal dysplasia, ectodermal damages, are subjective apohidrosis, nail dysplasia, sparse hair, throat abnormalities, and also we can find cleft lip or cleft palate, split hand, split foot malformation, with or without syndactyly. The last two conditions, SHFM4, split hand, foot malformation type 4, and orofacial cleft 8, that is isolated cleft lip, the cleft palate, are isolated form with no combination with other signs in organs, tissue, or other systems of the body. So the anomaly occurs in isolated form. Split hand foot consists in hypoplasia or in absence of the fingers and metacarpal of the hands, and of the central metatarsal of the feet, which have so-called Kela appearance. In this table are reported the main clinical feature of the various conditions of TP63-related disorders. Findings associated with a single phenotype include l'ankyloblepharum filiforme andatum, that are tissue strands that completely or partially fuse the upper and the lower hyalis, and also skin erosion, especially on the scalp, associated with areas of scarring and alopecia, hypospadias in male patients, trismus of the yaw, and excessive freckling on the skin. As regards, in particular, EEC syndrome, the clinical manifestations are typically present at birth. And let's see them more in detail. Ectodermal defect typically manifests as silver blonde hair that are coarse, dry, sparse, thin hair. Eyebrows and hyalis are sparse. Hair changes become more obvious with age and are seen in 60–80% of individuals with EEC syndrome. Dry skin and unique pigmentary skin changes, but erosions are not present in this form. Is commonly reported the nail dysplasia. Dental anomalies, including oligodontia, a reduced number of teeth, with a conical shape that is typical. Enamel defect also become evident during childhood and during adolescence. And xerostomia is the dry mouth and the subjective decrease in sweating capacity that is typical. So cleft lip, with or without cleft palate, is present in the major part of patients, 16–75% of individuals. Is bilateral in half of cases, and there is a wide variability in the severity of the cleft. Clefting can include, in fact, submucous cleft palate only, or a cleft of the soft and of the hard palate only, or only cleft lip, or combination of cleft lip and palate. Limb anomalies are seen in about 17–19% of cases. Also here, a wide variety of limb abnormalities are reported, including ectrodactyly, split hand, split foot malformation, syndactyly, oligodactyly, and digital duplications. 16% individual affected have a tetramelic involvement, which means in all 4 limbs. let's see in the eyes. Other frequent signs are absent lacrimal puncta. Is reported in 19% of individuals affected and results in tearing, blepharitis, dacryocystitis, keratoconjunctivitis, and photophobia, and often led to corneal ulceration and scarring. There are also, but occasional, blepharophimosis, and occasionally we can try genitourinary malformations that are reported in 14% of the patients, that may include hypospadia in males and developmental abnormalities of kidney and urinary collecting system. The TP63 related disorders are inherited in an autosomal dominant manner. It means that each child, male or female, of an individual with a TP63 related disorder has a 50% of chance of inheriting the pathogenic variant and with it the parent syndrome. Approximately 30% of individuals affected have an affected parent, and approximately 70% have a de novo pathogenic variant, so no parent affected. Genetic test is established on blood sample of the patient by a sequence analysis of TP63 gene, looking for point mutation of the coding gene sequencing. If no pathogenic variant is found, sequencing test is followed by gene target deletion duplication analysis. And so prenatal and pre-implantation diagnosis for pregnancies of increased risk is possible just if the pathogenic variant of the family is known. And I thank you for the attention.
Thank you, Savina, for your presentation, very interesting. Can you stop screen sharing, please?
Yes, sure.
Okay, thank you. Okay, perfect. And so many questions have arrived. And the first one I would like to ask you is, how many variants of EE syndrome are there? Are the symptoms of the different variants the same or different?
Okay. The clinical descriptions I have brought you correspond to the classical EEC syndrome, the type 3, that present some degree of genotype-phenotype correlation and present a very wide variability in clinical expression. That is a characteristic of autosomal dominant genetic diseases. The other variants of EEC syndrome correspond just in one EEC type 1, which show associated clinical features such as auricle malformation and middle and inner ear duct malformations. The gene responsible for the EEC type 1 form is unknown, but we know just it is located on chromosome number 7 on the long arm at locus 2-1, but the specific gene is not known. And EEC syndrome type 2 actually does not exist anymore. The other questions, from a genetic point of view, EEC syndrome and ectodermal dysplasia have a different genetic nature. and the difference is exactly in the gene involved. Ectodermal dysplasia depends on the mutation of more than 50 different genes, and also the pattern of inheritance is different because maybe autosomal dominant, like EEC, but also autosomal recessive and X-linked form with different risk for the son of a parent affected. And the treatment regard symptomatic treatments can be the same when we refer to treating symptoms. But when we refer to gene-targeted treatments, that cannot be the same because they are designed on a specific gene.
Thank you very much for your answer. And now I pass the floor to Dr. Tricarico, a psychologist who works with our association, and we talk about psychological support to the family.
Thank you, Sabina, for the answers. I remind you that you can send us your questions in the comment box below. As long as the director connects to the next speakers, I introduce him. Now the floor to Dr. Michele Tricarico, psychologist that collaborates with our association from 2 years, giving support to the patients and their families.
Good morning, I'm Michele Tricarico and I work as a psychologist. I work in different fields of psychology. One of these are rare diseases. Today, we'll talk about quality of life. In particular, we'll focus on people with rare disease. So we'll take a deeper look into the subject of communication and we'll analyze intra-family communication and the communication between a rare patient and healthcare professional. We'll try to provide to healthcare professionals some starting points to study in more details this subject and some practical advice to manage relationships with rare patients. It's not easy to define quality of life. Through time, its explanation has been changed several times. In my opinion, the most suitable definition has been provided by Quality of Life Research Unit in the University of Toronto. The University of Toronto defines quality of life as the degree to which a person enjoys the important possibilities of his or her life. Possibilities result from the opportunities and the limitation each person has in his or her life and reflect the interaction of personal and environmental factors. The definition is the result of 3 life domains: being, belonging, and becoming. Being refers to physical being, such as a physical health, nutrition. It refers to psychological being, such as a psychological health, feelings. And it refers to spiritual being, such as his personal values or spiritual beliefs. Belonging concerns relationships in social environments, such as family or friends. And the community environment, such as employment, income, community activities. Becoming pertains to practical becoming, for instance, domestic activities, volunteer activities, to a lesser becoming, free-time activities, and to a growth becoming, for example, activities that improve knowledge and skills. Now I'm going to describe the previous life domains, referring to rare patients and their different age range. Being the leading feature of rare patient is the self-acceptance of his condition. In such a situation, acceptance means facing a new challenge. Challenge is not to be impassive in facing the event, but changing together with it. Meanwhile, by recognizing the pain without losing the ability of being proactive. Now, children didn't develop the accept rare disease. For this reason, acceptance must be taught by family, parents. An adult receiving a rare disease diagnosis has to face the complexity and the difficulty to replace his present and his future to join acceptance. A non-person has to deal with difficulties of rare disease and old age. For belonging, for a rare patient, the most important features of belonging are being accepted by others with his own difference, and be active part in their own community, not being endured. As a child, a rare patient will be able to perceive belonging only if his family and his peers will accept him. As an adult, a rare patient will be able to feel belonging if he'll have a friendship or love relationships. As an old and retired man, he will be able to feel belonging if he'll have an active life. That's to say, sharing and participating in activities that can stimulate his life and slow down cognitive impairment. And now becoming. Referring to becoming dimension, the most important features for rare patients are the possibility of being active in our community and the possibility to adapt himself to the inside and outside change. A child will have the possibility to being an active part in the school and the leisure environment. Clearly, he will have to be held up by adults. An adult will have the chance to work by sharing his potentialities with the social and the work community. An old man will have the chance to being important in his community and family. Now we talk about communication. Man is a social being that lives and communicates in different social environments. For a high level of quality of life, communication must be as good as possible. This is an absolute must for people with rare disease. Today we are going to analyze the two features in the communication context: intra-family communication and the communication between rare patients and healthcare professionals. Family resilience is the absolute starting point to obtain a good intra-family communication. Resilience is the ability to deal with different contextual and developmental challenges. It is not a fixed idea, but it's a continuum. Now, the following four features allow to understand if there is a good level of family resilience. Being very close in facing hardships, being tolerant in dealing with different feelings, flexible rules but steady organization, and informal support network. A family can be very close in facing hardships when solidarity is the basis of the family relationship. Solidarity means that the concept of the rare disease can be shared in the family, and all the relatives are dealing with the hardships. Being tolerant with different feelings means that everyone in the family is able to express his feelings and to accept that of others. We can talk about the flexible rules and steady organization when family is able to adapt to good or bad changes. Every family member can be adaptive depending on his or her capability. A family that lives in good informal network, social association or friend, will be able to join a good level of family resilience. And now, communication between rare patients and healthcare professionals. In my experience as a psychologist, this kind of communication is often lacking. Rare patients and these families are confident in healthcare professionals, from diagnosis to therapy. They express their feeling and their hopes to healthcare professionals. For this reason, good communication is essential. Active listening is the best technique to achieve a good communication between rare patients and healthcare professionals. Active listening is fundamental for good communication and is the capability to listen to what we are hearing and at the same time understand another point of view. Active listening allows for high communication efficacy. This improves therapeutic alliance. It means that rare patients will listen gladly to healthcare professionals and will understand the recommendation in better way. And now some important rule in active listening. Focusing. While the patient is talking, the professional has to try to understand what he is telling instead of thinking about the answer to give him. Not judging. Many times, healthcare professionals judge patient behaviors. For example, they say, "You are wrong." Don't stop the patient. The patient will not feel understood and will not accept advice because he'll think that the professional will not have understood everything. Summarize what it has been told. After the patient speaking, trying to summarize in order to be sure to having understood and possibly asking for more explanation. For example, by saying, "If I'm right, you are telling me." According, by using words that patient will certainly understand and trying to use the same attitude. Pay attention to the emotions. A good therapeutic relationship is based on trying to understand how the patient feels. Through not-verbal communication too. After talking with the patient, it can be useful to ask him how he is feeling. Except, while patient is talking, nodding or saying something comfortable can be useful to make patient more accepted. Respect the silence. Some silence can be useful during communication because it allows the patient to reflect about what he has heard. Trying to fill silence can reassure professionals, but at the same time can make confusion in patients. And then maintain eye contact. Some research papers show that maintaining eye contact for almost 16–17% of the time may improve relationships. Okay, now I'd like to conclude my presentation giving you my contact information and thanking you for your attention.
Thank you, Michele. I remind you all that our medical staff is always available also for the psychological support. The next intervention, recorded especially for us, a very special person for me, the first person who took me in her arms at the birth, a friend as well as an excellent doctor. For this reason, I am honored to leave the chant to her, Dottoressa Beatrice Della Barba, head of the birth department of the Padova Hospital, pediatrician and child neurologist.
Buongiorno a tutti, sono Beatrice Della Barba e sono con voi con il cuore. Sono felice che sia stata fatta questa breve registrazione perché per me è un grande dispiacere non essere con voi in questo momento perché ci tenevo tantissimo. Quindi ci vediamo via web, ma anche questo qualche volta serve. Io ringrazio tantissimo per l'invito che ho avuto e per me è un grande onore far parte di questo gruppo. C'è anche un motivo molto forse personale che però vorrei citare non solo per il suo aspetto personale, ma perché è un aspetto che diventa importantissimo quando si ha a che fare con dei bambini che possono avere dei problemi e soprattutto quando parliamo di bambini che possono avere delle malattie pesanti e rare. Io ho conosciuto Giulia, che è, io dico sempre che è uno tsunami, perché ha una carica di energia e di capacità che veramente è invidiabile. Ho conosciuto Giulia quando è nata, ma ho conosciuto anche la sua mamma quando è nata e è successa una cosa molto particolare che vorrei raccontare. Non molto tempo fa ci siamo incontrate dopo tanti anni, dopo quasi 20 anni e io e la mamma di Giulia ci siamo abbracciate con le lacrime e questo abbraccio commovente che io vi racconto vuole sottintendere non solo un affetto che ci lega da molti anni, ma vuole sottintendere forse qualcosa che io vorrei sottolineare e che vorrei anzi enfatizzare, e cioè se c'è un ricordo di questo genere tra me e la mamma di Giulia e poi tra me e Giulia non poteva ricordarsi di me. Questo è legato al tipo di comunicazione che c'è stato tra di noi, che c'è stato tra me e la mamma di Giulia. E se c'è un bel ricordo vuol dire che c'è stata una buona comunicazione. Ebbene, io sono veramente convinta, ma dal profondo del cuore, che la comunicazione, il primo colloquio che un medico o una persona fa con i genitori e con la mamma di un bambino che nasce con dei problemi, non debba essere legato alle caratteristiche personali, umane di una persona, ma sia piuttosto debba essere il risultato di una formazione specifica. Perché il primo colloquio che si ha con questi genitori, questo vale per tutti i genitori, ma a maggior ragione per i genitori che hanno bimbi con dei problemi, deve essere un must, deve diventare un must. La comunicazione deve essere fatta in modo ottimale, perché solo così potremo avere la fiducia di quel genitore, solo così potremo creare un rapporto positivo con quel genitore e poi avviare una presa in carico della famiglia, non solo della mamma e del bambino con questi problemi. Ecco, io credo che il problema della comunicazione e della formazione sulla comunicazione sia prioritario quando abbiamo a che fare con dei bambini con problemi, anche se dovrebbe essere, secondo me, doveroso sempre. Continuerò per sempre a complimentarmi per il lavoro che state facendo e anche per tutte le collaborazioni che credo sia molto positivo creare, e qui oggi vediamo la dimostrazione pratica di questo. Io credo e mi auguro che questo non sia, che questo sia un arrivederci a presto, e rispetto anche a quanto dicevo prima, credo che sia importantissimo l'aspetto della multidisciplinarietà, perché più lavoriamo insieme, tra tecnici, tra persone informate, tra persone coinvolte, più questa cosa darà i suoi frutti. Io credo che Giulia sia straordinaria, perché Giulia sta portando avanti, certo, anche la sua mamma, ma ha portato avanti da sola qualcosa che invece forse il mondo avrebbe dovuto portare avanti per lei. Perciò avrà sempre il mio appoggio, e io credo che dovremo dare un grande appoggio da parte di tutti a questa associazione e al lavoro che in questa ottica viene svolto. Buon lavoro a tutti.
Dear Beatrice, we hope to see you again soon. So thank you for your attention. The first part of our webinar is finished. We hope to see you again at 2:30 p.m. for the second part, Eyes and Skin. So good lunch for the Europeans and good breakfast for the Americans. See you later. Giulia.